So you want to know how a GMO is made. You're not alone. It's one of the top questions about biotechnology. We'll use the story of the papaya in Hawaii to walk you through one of them. Step 1. Determine whether genetic engineering is the most effective way to solve a plant's problem, such as a disease or damage from pests. A powerful new tool, biotechnology, was now available to fight back. Farmers and researchers turned to genetic engineering beginning in 1985 for help. Step 2. Identify the gene or genetic material that could solve the problem, like providing resistance to the disease or pest, and study the genetic makeup of the plant that needs the trait. Researchers and farmers had to find a way to make the papaya plant resistant to the deadly virus. PRSV resistance was the genetic trait needed to solve the problem. Step 3. Copy the trait from a donor organism and implant it into the plant's DNA. Researchers isolated a PRSV gene from the virus itself. Then they needed a way to get the genetic material into the papaya plant's own DNA. Researchers deployed a commonly used technique in biotechnology called transformation, a process by which a copy of genetic material from a specific trait is inserted into cells. Material made the plant resistant to the ring spot virus, just as a flu shot makes people resistant to the flu. The researchers were able to successfully transform 17 plants. These plants contained this gene from the virus and produced a protein from the virus that helped the plants become resistant to the PRSV infection. Step 4. Plant the new seed and test it. The final step was to plant the seeds and carefully test the resulting plants. After several years of testing, it was determined that this line performed well on the farms, was safe for people to eat, and was of commercial quality.